Oh yeah,
Hey everyone. It is my great pleasure to introduce Michelle Zhu for her appraisal seminar. It's also my great pleasure to advise Michelle. She started is in her second year. We all do our second year now. She came to me after doing an undergrad in EB, working a lot in Luke's lab on some cool and null questions. Starting at the beginning of her PhD, she completely switched gears and started working on sand dollars. I was pretty gratified. Wait, there's like a sound coming. Oh. Is this someone that's stuck? Okay, so I'm easily distracted. I mean, it doesn't bother me really.
I just muted. Okay, I just muted the whole computer.
Okay. Glad we got that sorted. Anyways, yeah, and completely switched, and I was super gratified by it. I was because I think it's a cool system. I'm pretty interested in econoids generally, and Michelle has just sort of taken Dendraster specifically and fallen in love with them and just run off in a million cool directions, I think, in exploring questions with these kind of funky little guys. So I don't know, without further ado, I could ramble probably for a long time about Michelle's cool work, but I'll just let her do that instead. So let's welcome Michelle.
Okay, right. My computer is muted and it shall stay that way, and I hope that nobody's trying to talk to me for whatever reason. Let me set a timer for myself so that I can feel at ease. Okay. Okay, thanks everyone for coming. I really appreciate it. So I guess I'll just start then. Variation is everywhere, both within species and between species. And as evolutionary biologists, we're fundamentally interested in studying this variation, and we can do so from a variety of different angles and ask many different questions. One question that we often ask is, how does variation change within species? This type of evolution can generally be called anagenesis, or evolution that occurs within a lineage, and many different processes are at play in this context, including directional selection toward a new optimum, like in these peppered moths, or stabilizing selection that culls extreme variance around a consistent optimum. You can also have selection that favors the maintenance of variation to exploit diverse resources, for example, and you can also have evolution through drift. But of course, the variation that we see within species is the ultimate source of variation between species as well. But how does it get from point A to point B? How does variation get inherited in newly forming species? And this type of evolution we can generally call cladogenesis, or branching that generates new lineages. And again, many different processes are at play here. The variation that the descendant inherits will depend on the sample of variation that they get and on the population size at speciation as well. And also the presence of natural selection, if the descendant inhabits a new environment relative to the ancestor, will also determine what variation they get. So these two dynamics are both describing variation once it has arisen, but it can't tell us where variation comes from in the first place, which is a whole other set of questions that we can ask. How do new variants arise, and what forms are possible? To understand the actual origins of variation, we have to try to understand how the phenotype is constructed, and this depends on things like pleiotropy, epistasis, all the intricacies of development and construction, etc. And because of this, not all conceivable variation is possible or even equally likely. For example, some traits will tend to co-vary, like in these butterfly eye spots, the color composition tends to change in the same direction, and it's harder to break that correlation. Another way that development can have big impacts is that some relatively small changes, like changing the expression of one gene or the timing of one event, can have cascading effects across an entire appendage or the entire body. And this can be an important source of novelty. So these questions have been around since the beginning of evolutionary biology as a field, and in our ongoing efforts to answer them, fossils have been and continue to be a really essential resource because of the unique information that they can provide. Fossils are the only direct evidence of past variation. So in these carrion snails, some of the polymorphisms you can see in recent populations are also found in Pleistocene fossils. They are also the only direct evidence of change through time, especially geologic time. Like in this case, you can see how the relative frequencies of these three morphs changes over 4 successive ammonoid species. If you have a decent fossil record, you have a good chance of sampling ancestors or taxa that are very similar to them. whereas otherwise you'd have to reconstruct the other states, which would be, questionable accuracy. And finally, fossils can tell us about when species originate, when they go extinct, and patterns of temporal co-occurrence. So I hope that I've thoroughly convinced you about how awesome and important that fossils are, because this leads me to the fossils that I work on, which are Dendraster. Dendraster are sand dollars, which are a type of highly flattened sea urchin. Dendraster are unique among all sand dollars for possessing this eccentric morphology where the apical system, which houses the reproductive and respiratory organs, is shifted out of the center or eccentric to the posterior. Most sand dollars are deposit feeders that lie flat in the substrate, but this eccentric morphology allows Dendraster to stand upright in the sand as well and access a completely new niche as suspension feeders. They also have a really extensive fossil record because nature has blessed sand dollars to be really fossilizable because of their rigid skeletons, flat shape, shallow marine habitat, and gregarious nature. Their fossils are so abundant and well preserved that we can trace the spread and evolution of this group from their origin in the late Miocene of California to their present day distributions from Alaska down to Mexico. And because of these great fossil record, we can also look at variation within Dendraster, such as variation in overall 2D outline or variation in the height or thickness of the test, which is the skeleton. And we can also see how variation changes over time, like in this tubercle polymorphism, tubercles are the little nubs that spines articulate onto. Over the course of 400,000 years, the relative frequencies change until one of the morphs is lost entirely in this species. So for my thesis, I'll be using this really great fossil record to study variation across 3 levels. In chapter 1, I look at variation across the phylogeny. In chapter 2, I look a little closer at the dynamics of variation through space and time within populations. And in chapter 3, I try to take a look at the origin of variation through a developmental lens. So for chapter 1, for any clade-wide analysis, we need a phylogeny. And as it stands, there's no formal phylogenetic analysis for this group yet. The best thing that we have was by Q in 1920, and Q was a taxonomist who proposed relationships based on morphological, geographic, and stratigraphic information. But this was over 100 years ago, before the phylogenetic theory as we know it even existed. So I hope to bring our understanding of the relationships up to speed within a modern analytical framework. And there are a lot of challenges to reconstructing relationships in this group, especially that there's a lot of intraspecific variability and a lot of overlap between species, which has confounded many researchers, including Raup, who did his early work on echinoids, on Dendraster in particular. And as I said before, variation can be inherited by new species. And in fact, shared polymorphisms across clades is pretty common in phylogenetic datasets. But despite the ubiquity of this shared polymorphism, most phylogenetic methods are not equipped to deal with it. Most of the time, polymorphisms are just ignored, especially in morphological phylogenies where you can choose what characters to code and how to code them. And if they're incorporated at all, they're usually just treated as uncertainty and integrated over. But this is a shame because the shared polymorphisms are not just a nuisance, they can actually be informative, and they can tell us about evolution, especially in younger clades like Dendraster. So the inadequate treatment of polymorphism limits the inferences that we can draw and ultimately the processes that we can study. And moving forward, we should work on incorporating and developing new methods that can work explicitly with polymorphism, which leads me to my chapter 1, where I attempt to reconstruct relationships in Dendraster using polymorphic morphological characters and stratigraphic data. And I also investigate how polymorphism is distributed throughout the clade and potential patterns in its inheritance. So the data set I have includes 11 species, 3 extant and 8 extinct, which is more or less all of the species barring taxonomic uncertainty. And I coded 35 morphological characters using images of museum specimens that I took at the Natural History Museum of Los Angeles County and the UC Berkeley in Paleo collections. And I also incorporate stratigraphic data, which I obtained from the literature. So the model of morphological evolution that I use is called the mutation loss cladogenesis, or MLC model, and it has 2 components. The first component is cladogenesis, where in an instant process, the descendant inherits some subset of its ancestor's variation. And the other component is the mutation and loss, which are encapsulated by anagenesis, which is a time-dependent stochastic process where the lineage can gain or lose states according to the matrix shown here, transitioning from the unobserved set of inherited states to the observed states at the end of the taxon's duration. Moving on to stratigraphic data. We incorporate stratigraphic data because it can actually provide us with phylogenetic information as well. For example, here, just looking at these stratigraphic ranges and with no other information, you see A appears before B and C. So you might start to speculate, maybe A is ancestral to B and C, or maybe it diverged before B and C did. And we can formalize these intuitions using models. For example, here it's just a simple Poisson model of some constant rate of fossil preservation. And the behavior of this model is essentially that it tries to minimize the unsampled time implied by a tree topology. So here there are two topologies with the exact same set of stratigraphic ranges. In topology 2, there's more unsampled time implied, here highlighted in red, which means that topology 1, instead, where A diverges first, would have a higher likelihood. So the overall tree likelihoods are just the joint likelihood under both topological and stratigraphic models. I find a tree that maximizes this joint likelihood, and I score the trees using the FAK information criterion. So what does the best tree look like? Okay, on the x-axis is time, and moving toward the left, you're going into the past, and toward the right, you're going into the present. This is the tree, and on this tree, the dark gray bars represent sampled time, we'll be acting out fossils, and the light gray bars represent unsampled time implied by the tree topology. And the dashed lines represent connections between ancestors and descendants. So there are some similarities to the Q tree. For instance, there's a clade here that contains Dendraster, Venturaensis, Diegoensis, and Eccentricus. And there's also some differences, like Dendraster ashleyi is not recovered as being ancestral to this group and is instead its own offshoot off of Dendraster gibbsii. And there's another similarity in this lineage leading from Dendraster gibbsii to Coelengaensis to Perroni. So overall, it's heartening to see that there is some statistical support for these previously hypothesized relationships, and moving forward, I can use this great robust tree as a foundation for my next future chapters. As for variability across the clade, I look at it with a very simple metric of variability, which is just the number of polymorphic characters divided by the total number of characters, so the proportion of polymorphic characters. Plotting it on the tree like this, where bar width represents the degree of polymorphism, you might be able to see a trend of decreasing polymorphism as you move toward tipward taxa. If you visualize it another way, here on the x-axis is the amount of polymorphism in the descendant, and the y-axis is in the ancestor, and this diagonal line is the identity line. Granted, there are not that many points, but in most cases, the ancestor is more polymorphic than the descendant. This is consistent with many different mechanisms, including speciation, sieving, ancestral polymorphism, or population bottlenecks at the beginning of a species and subsequent fixation. Could be adaptation from standing variation that culls variance, and it could also be that more variable species have a greater rate of speciation. It could be something else entirely. But I hope that I could dig into these patterns more quantitatively in chapter 2, where I look at the dynamics of variation through space and time within populations. So I've spoken a lot about time because of course evolution happens through time, but I will introduce the idea of space and geography in particular now. So variation and geography are deeply intertwined. Widespread species are typically more variable. This is partly a raw numbers thing. A larger area supports a larger population that harbors more population. Also because of geographic variation, and across a wider range, there's more possible heterogeneity. This variation can occur due to natural selection in a heterogeneous environment, but can also occur purely due to drift and population structure. And of course, we all know, and I'm sure we all know, that geographic differentiation is an important precursor towards speciation, in particular allopatric speciation, and that this effect might be especially rapid in small peripheral isolates that are evolving faster through drift and experiencing more extreme selection in this more extreme environment. So with these ideas, we can predict certain geographic and phenotypic patterns involved in speciation, and we can study them using extant populations, or try to, but it's difficult because populations evolve and change and ranges shift, so that the phenotypes and ranges of today may not represent what they were at the actual time of speciation. But fossilized populations can allow us to try to capture the interplay between geography and evolution more close to the time of speciation actually itself. So in chapter 2, I hope to investigate evolutionary changes within populations and at speciations, especially for particular variables: geographic range location, geographic range size, phenotype or average phenotype, and phenotypic variability. And I also ask if we can identify patterns that are consistent with what you would expect in cladogenetic and anagenetic evolution. So the methods I propose to use will once again include my museum specimens from LA and Berkeley. There are 2,528 specimens that were imaged in total. And when sampling, I tried to get localities that were representative from across species geographic and temporal ranges. As for my temporal data, most of the specimen localities have their ages resolved to the stage or epoch level. And if I want further resolution, then I can look at the local stratigraphic literature where they further subdivide formations into local sections or zones. For my geographic data, I have a very tentative map based on the lat-long coordinates of the Dendraster fossil localities at these museums. And using these occurrences, I can quantify a geographic range using convex hulls. And finally, to quantify morphology, I will be using landmarks to obtain linear measurements. This is a sample of the landmarks and measurements that I could use. Some of the measurements include things like overall length from front to back, width, degree of eccentricity, the lengths of the petals, things like that. And to quantify the mean phenotype, I will use centroid, which is a multivariate mean, and variability I can quantify with total variance, the sum of the individual variances for each of these phenotypic traits. So with all these measurements and metrics in hand, I can then compare ancestor and descendant pairs as specified by the tree from chapter 1 and ask, how do geographic range area, the centroid of the phenotype, and the total variance of the phenotype change across these events? So I'm going to show you a bunch of graphics. In all of them, time is on the y-axis. These ellipses represent geographic ranges. The color of the ellipse represents phenotype, and the ancestor is on the bottom and the descendant is on the top. So we can imagine maybe there is a range shift, or maybe there is not a range shift. We could ask if there is a change in range size, whether it decreases or increases, or maybe it doesn't change. We can ask if there's a change in the average phenotype and also ask how large this change is. And finally, we can ask if there's a change in variance as well, and again, how large this change in variance might be. We can also look at certain predicted evolutionary associations between morphology and geography. For example, we can ask if a decrease in variance accompanies a decrease in range size, as you might expect if the more restricted range is less heterogeneous or if they're losing more variation through drift. You can also ask if phenotypic shifts are associated with range shifts, as you might expect if there is natural selection in this new environment that they've colonized. And finally, we could attempt to use this data to quantify or identify cases that more resemble anagenetic versus cladogenetic evolution. So I've collated some predictions together from literature. So in anagenesis, you may expect to see a range shift or not, and range size may or may not change. And we know that range sizes can shift throughout a species lifetime, but I don't know if there's a systematic change that you can expect. You could also expect more subtle morphological shifts or stasis and a similar level of variance in the ancestor and the descendant, because probably they're responding to a consistent selective pressure that is not changing a crazy amount, and that mutation selection balance would keep their variance more or less consistent. Cladogenesis, the expectations are a bit different. You might expect to see range shifts if allopatric and peripatric speciation are involved, which most of the time they probably are. You might expect that the descendant ranges are smaller than their ancestors. Again, if small populations are especially important for diverging into new species, or if you just fracture the ancestors' population to get the new range. You might expect to see more dramatic morphological shifts and a reduced variance in the descendant. Again, because if they are evolving more quickly due to drift or experiencing more strong selection in a new environment, these could result. And if we can identify these cases that are more like anagenesis or cladogenesis, we could ask about the relative contributions of evolution at speciation versus between speciation events to the overall patterns of the clade. And we can ask about the different contexts in which these modes of evolution occur. So I hope that with chapter 2, I will be able to test predictions generated from theory about how geography and morphology evolve in natural populations over geological time at and between speciation events with an unparalleled level of resolution. And I think that thoroughly investigating and taking stock of evolutionary changes and their contexts within a clade where it is tractable is a really great opportunity, because most of the time this might be prohibitively complex or obscured. And this would be a great chance to dig into the processes that are acting within populations and at the interface between population and species. So in chapter 3, I will talk briefly about how I want to look at the origin of variation through a more developmental perspective. And one oft-cited and particularly fruitful source of variation that people talk about is heterochrony, or evolutionary change in the timing of ontogenetic events. So a classic example is that the axolotl, instead of becoming a terrestrial adult, retains a lot of its juvenile aquatic traits and looks like a juvenile or a child in that way. And heterochrony often has large morphological and or ecological effects, which leads people to speculate that it might be an especially important source of novelty and evolutionary change. And in fact, Dendraster counts as one example of heterochrony with really big ecological effects, because it's speculated that the eccentric morphology arose through heterochrony. So in related sand dollars, they start out by developing quicker in the anterior section and then later in life switch over to developing more posteriorly, which evens them out to be symmetrical. But in Dendraster, if you never switch away from the first growth mode, they just keep growing anteriorly and become more and more eccentric. And recall that this relatively simple change has had profound impacts on their ecology and allowed them access to this entirely new lifestyle. Others have also proposed that other species within Dendraster appear to be pedomorphic, suggesting that this might be a more general phenomenon in the group. And there are a lot of other morphological differences between the species that could be understood through heterochrony. For example, there's a large difference in specimen size or species like body size. Like there are some really big hand-sized ones and some really tiny ones that are the size of a lentil. And there are some species that are really eccentric, some that are less eccentric, some that have larger tubercles or smaller tubercles, are flatter or more inflated, some that are more pentagonal shaped or rounded, etc. And all of these are traits that vary over the course of autogeny as well. So I think that heterochrony would be a fortuitous place to start looking to try to understand where these morphologies come from. So in chapter 3, I hope to determine whether heterochrony can explain the evolutionary divergence we see across Dendraster species, and also investigate potential correlations between heterochrony and ecological variables. One approach that we can use to study heterochrony in fossils is to map out shape changes through ontogeny, and we can do this by regressing shape variables onto a proxy for age, usually size in fossils. And from this, you can get an ontogenetic shape trajectory, which is just the trajectory through shape space that a species or a taxon traces as they are growing into adulthood. And then using this, you can compare it to the descendant morphologies. So that descendant morphologies that lie close to this trajectory you can attribute them to heterochrony. They could be easily generated by heterochrony, whereas morphologies that are far from this trajectory are less easily attributable to heterochrony, and you probably have to invoke some other type of change to explain them. As mentioned, I'd also like to look at some ecological information, and here are a few potential paleoenvironmental variables, such as water temperature, sea level, nutrient concentrations, substrate grain size, and environmental instability. All of these could potentially select for the morphological and life history changes that heterochrony can produce. For example, grain size is associated with feeding and with spine size and tubercle size. And another is that environmental instability can select for quicker maturation time, which is an effect that heterochrony can produce. Overall, with chapter 3, I hope that we can have a better understanding of evolution form in Dendraster, specifically where new morphologies come from and why they persist. And overall, in general, heterochrony is believed to be a really common phenomenon, so that understanding it in this group can shed light on how developmental processes generate morphological and ecological novelty across more diverse taxa as well. So to conclude, with my dissertation, I'm hoping that I can work closely with the system and really understand their natural history, their ecology, their development, to ultimately get a deep understanding of their evolution. And it's kind of rare to have such a great fossil data set that reaches through geological time and across species, but also has enough data that you could look at what's going on at the population level. And ultimately, I think that the evolutionary insights that we can get from working with this group could apply to processes that are acting across the tree of life. And so with that, I'd like to thank you all for being here and listening. And I'd like to thank everybody in the lab for supporting me and who let me into the museums and took photos with me. And I will take any questions. Way fast.
The first one was from your chapter 1 section.
Okay.
Where you said that the width of the bars indicated the amount of polymorphism or something, and so there was less in the younger than the ancestors. And I was wondering, does that just have to be true based on your model? If there's two related younger taxa that have different states, then doesn't the model say that the ancestor has to have both of them? Or did I misunderstand something?
Okay, so in this model, you can have direct ancestors, as in like a taxon in your sample could be a direct ancestor. So that it wouldn't necessarily always reconstruct an ancestor to be more polymorphic. And in this case, I use the level of polymorphism like in the data set, which is assumed to be like at the end of a duration. And so even though during cladogenesis, they can only lose states, over anagenesis, they can gain a state, so that ultimately that will result in the data that we see here. That makes sense.
So the pattern you saw is not.
I don't think it's an artifact of the,
And my second question was about in your second chapter where you're looking at patterns of phenotypic variation and differences in phenotypic mean to infer something about basically the genetic basis of these changes. Just wondering, obviously, that it's not a one-to-one